Yes, go live. Yes, live started. Thank you very much.
Yes, Sundar, thank you so much.
Okay, so
Yesterday also I have tried. Because we cannot test to go live and all. No. Once you click to go live, then that link will expire.
Yeah.
We cannot use the same link again. So that's the
Yes,
problem. Okay, now thank you so much.
Yes, so I mute my camera and the microphone, but stay monitoring. So if you have any trouble, please feel free to contact me.
Thank you so much.
Good luck.
Yeah, thank you.
மொபைல்ல கனெக்ட் ஆகிறதுனால ரெண்டு சவுண்ட் வருது.
இந்த இது வேற இருக்கு.
வேற யாராவது ஜாயின் பண்ணிக்கோங்க.
ஓகே, அது கனெக்ட் ஆயிருக்கு. இங்க மியூட் பண்ணாததுனால ஹலோ
ஹலோ
அதான் லைவ் இதுல
சொல்லுங்க சார்.
என்னுடைய தான் வருது.
என்னோடது வரலையா?
ஸ்கிரீன் லேஅவுட் ஏதாவது மாத்தணுமா? ஹலோ
ஹலோ
வருது, இப்ப வருது. அதான் லைவ் இதுல
சொல்லுங்க சார்.
என்னுடைய தான் வருது.
என்னோடது வரலையா?
ஓகே, இப்ப நீங்க பேசினா வருது.
கிளியரா இருக்கு. நான் பேசும்போது என்னோட வீடியோ வருதுங்க சார்?
கரெக்ட்.
ஓகே.
கொஞ்சம் டிலேவும் இருக்கு.
டிலே இருக்கு.
லேசா இருக்கு. வருது, இப்ப வருது.
ஓகே.
ஓகே பேசும்போது வருது. இவர் கனெக்ட் பண்ணிருக்கார் போல.
கனெக்ட் பண்ணிருக்கார் சார். கனெக்ட்.
சரி, நீங்க பேசிங்க இதுக்கு அப்புறம்.
ஓகே சார்.
நான் மியூட் பண்ணிடுறேன்.
ஓகே. Hello.
Hi.
Hello, sir. Good morning, sir.
Good morning. How are you?
Fine, sir.
Nice meeting you, sir. I'm Dr. Sheila John from Shankar Nethralaya.
I'm Pramila, sir.
Oh, thank you very much for the invitation.
My technical person who's going to help us. Thank you.
Thank you, sir.
Thank you. So how many minutes do I have? Is it 15 or is it 20? I couldn't.
No. 15 minutes you can talk, sir.
Yeah.
We have even 5 minutes of discussion.
Okay.
Question and answer. Then we'll have discussion later. So your talk is will be for 15 minutes, sir.
Come on, 15 minutes.
You will have to do 1 or 2 minutes of self-introduction.
Excuse me?
And then you have to do 1 or 2 minutes, 1 minute of self-introduction. Introduce yourself.
Oh, I see. Okay.
Because this is from the Asia Pacific region. This is a WHO center in Japan. Temtech, you must have heard of it.
Okay.
And they've established a WHO center there, which they're trying to do through video conferencing and do a lot of activities. So,
Why is it 52? What does this 52 mean?
then that is each conference, they conduct about 2-3 conferences a year.
This
The
is
number
the
of conferences they've conducted. It's a 52.
this is the 52nd
52nd conference.
conference. I got it.
They do 3 programs. They do something for engineers.
I see.
Because they want technical people to be very sound in doing video conferencing, YouTube streaming and all that. Otherwise, the interruptions will come. So that is done. That's one part of it. Then the medical wing group has different options. Each time I've done something, I'm an ophthalmologist. So I do a lot of telemedicine.
Oh, I see.
Ophthalmology.
Yes.
So that we have done various how to do DR screening, how to do cataract screening. Then we had something on general teleophthalmology. Then we have had it on uveitis. So we have a lot of connecting institutes. And then we do it.
I see. So you have this vast network.
Yeah, this is a network, and it's basically in the Asia Pacific region.
Oh, I see.
Today we have one hospital from Vietnam connecting. Sometimes Australia people connect. Then Asia Pacific. And some people are from India. So basically we have 3-4 speakers, and the delegates are from various connecting institutes.
I see.
Earlier it used to be in person, one place. They usually select each country in Asia specific region. We had it in India also in Delhi.
Oh, I see. Well, I've been to Delhi many times. So.
There's one person, Mr. Ghani, he's a technical person. He went for a basic workshop there. So each person goes in different directions. They have it in major cities in the state.
So how's life in India? How's life in India with this virus going on? So,
it's a mask all the time. I sometimes think whether I don't know whether I'll get corona or not. But due to the mask, I will get some problem because I'm wearing it all the time. Breathing, no? Oxygen is not going in sufficiently. So I'm sure I might get something due to that.
Especially when it's hot. It's
very difficult. So that's the only thing. It's very hard. And second, there is a lockdown. They close everything and then they release everything.
I see.
When infection spreads, no? That's what they do.
Lockdown is a real lockdown. It's really a lockdown. You can't go anywhere. Or is it
the lockdown is some amount like hospitals work. So we extend passes and we can come in. Then the police people work. The corporation works. It's only the other people who sit at home. The shops are closed. They'll have timings to open the vegetable shops and other food items. All other shops are closed.
Yes.
But is the infection controlled in Japan?
Well, I don't say it's controlled. I think this Delta strain, I think it's coming from India. It's spreading all over Japan. And we're having this Olympic with during the surge of this Delta strain. So
yeah, they say different strains come in, no? Yeah. You get used to a train and a variant comes in from another country. Some countries are completely closed. They don't allow outsiders in. That I think maybe is a safer option.
There's another strain coming from Peru. Another one.
Oh.
More serious.
Those countries are closer to you, no? Japan means that other US and North America and South America are much closer. And Australia also must be somewhere near.
Yes, Australia, when they have like one patient, they do lockdown. So okay, where's Nataraja?
He's joined, sir.
He's joined.
He's joining, or
he must be joining. Very busy person, sir.
He's joined.
Hey, good morning, Takeshi.
Good morning.
How are you?
How are you?
So yesterday, Dr. Sheila John was worried. What happened to Takeshi?
Takeshi.
So I said he's busy with the Tokyo Olympics.
Yes, attending the Olympics.
So I'm happy some Indians are winning some medals.
Oh, I see. Which games?
Wrestling, hockey, men hockey, women hockey.
Okay.
Then what is the other one? I think shuttle.
Okay.
Badminton.
Badminton, yes. Mm-hmm.
Yeah, and we had one Milkha Singh who recently died at the age of 90. He won the not Milkha Singh. What is his name? Who the runner in 1940 Olympics in Tokyo? He won the gold medal.
Gold medal. Wow. Running.
I think so. Even hockey India won the gold medal that time.
I see. Mm-hmm.
Who's the other runner, Pramila? One is Milkha Singh.
We don't remember. We don't remember, sir. Sorry. Speak first.
So Dr. Sheila John is a consultant, and she's a PhD in teleophthalmology in Shankar Nethralaya.
You were telling me about teleclinic, telemedicine. Yeah, telemedicine.
So we have started a Tele-Ophthalmic Society of India.
Oh, I see.
I'm the founding president.
You're
president of everything.
Yeah. That's what I was yesterday doing his biography. I've seen president of so many organizations. So many. I think to his name, president can be added.
Yeah. He's a great leader, man.
Leader.
Leader.
So we both are doing a big lab in Mumbai. And Takeshi, we got one just about half a mile from hospital. We have one college where there's a National Center for Biotechnology, and I'm actually doing an MOU. I'm going to shift that lab from that previous place where you came and keep it here. And there are a lot of PhDs working there.
Oh, I see.
And it's just walking to the hospital.
Oh, it's a walking distance.
Yeah.
I see.
And
so first it was very far away, but it was very nice building. And then you came a little bit closer and you're moving again.
Yes, now it's still walking.
Still walking.
To the walking.
Okay.
It's 9:31, Sheila.
Yeah, we'll start the conference.
Yeah.
Today's webinar is on ocular genetics, which is a growing field, and it's very important to identify genetic diseases in families, and counseling is to be done. We have very good speakers today. The speakers will do its self-introduction, followed by question and answer, and then discussion. The various speakers are Professor Dr. Natarajan sir from Aditya Jodh Eye Hospital, Mumbai, Dr. Takashi Ayavata from Tokyo Medical Center, Professor Dr. Madhavan from Shankar. Over to you, sir, Natarajan sir, for your lecture. So, your lecture, sir?
I am starting. So I'll just start. Thank you, Sheila. And we have Dr. Kyong there and Dr. Takeshi here. And also, so I'm happy to be a part of this. And thanks to Dr. Sheila Jan for organizing this ocular genetics. And I think very important. And we have Dr. Takeshi Iwata, who's the president of Global Eye Genetic Consortium, and he's also part of the very integral part of Japanese Eye Genetic Consortium, which is doing a great job with 35 universities attached to National Institute of Sense Organs. So my talk is on inherited retinal diseases and gene therapy, the current trends, and the background. The knowledge about inherited eye disorders has increased rapidly during the last 20 years, even though there are no global statistics available to understand the burden of visual impairment from genetic causes, but more commonly that genetic eye pathology represents a significant percentage of the causes of blindness globally. And Dr. Takeshi Iwata has created a Japanese Eye Genetic Consortium where they have a robust software for tracking phenotype and genotype, which India is lagging, but there are institutes like Shankar Nethralaya, Arvind, and L.V. Prasad are doing their own, and maybe Aditya Jodh too. And the retinal dystrophies, the clinically and genetically heterogeneous group of eye disorders affecting approximately 1 in 3,000 to 1 in 4,000 people worldwide, characterized by degeneration of different cell types within the retina. And retinal cell types involved in retinal dystrophies include the rods and cones. And retinal dystrophy, the diagnostic test, the electroretinogram, visual field study, optical coherence tomography, fundus photography, and all this will comprise the phenotype. And later, I think Dr. Takeshi will be talking about it, where once you are used to doing the phenotype and genotype, and then later we can know the characteristic, and probably without doing the genotype also, we'll be able to classify. But we need I think the best is to have the basics. And retinal dystrophies, the inheritance pattern can be autosomal dominant, autosomal recessive, X-linked, or mitochondrial. And more than 250 genes have been identified that can cause retinal degenerations. And this is 1980 to 2019, and you have the reference there. And also the genetic heterogeneity in retinal dystrophies, again published in 2015, where you see the cone rod dystrophy, the Stargardt's, and Leber congenital amaurosis, retinitis pigmentosa. And I think all put together, unfortunately, India will be the capital. And so I think that is the reason I'm glad the National Institute of Health and National Eye Institute, we had Dr. John Prakash as the founding president, and now he's the patron, which was started as an Asian Eye Genetic Consortium. Later, it has become Global Eye Genetic Consortium. So you can visit gegc.org, which Dr. Takeshi will talk. And the whole idea is in India, we are working individually with institutes, and we want to have our own, and that's what we are trying to create, because the number of patients in India are unfortunately quite a lot. And I think the government, the state governments have to take part in this. So the gene therapy, efficient and accurate genotyping is the key to developing possible gene-based therapies. And not only for gene therapy, but I think even to segregate the disease. And I think to say, okay, this part of this, what will be the prognosis and what will be the various decades of life, how they will be, is important to educate the patient. Because otherwise they think, and then the genetic counseling, everything will be helpful if you have a efficient and accurate genotyping. And gene therapy is effective only when the genetic defect is identified in a given inherited retinal dystrophy. And gene therapy is available is a hope for patients. And the main target is retinal pigment epithelium or photoreceptors. And the most common method to deliver viral vectors to the cells, intravitreal injection, subretinal injection, and access from the suprachoroidal space. And I'm glad as a vitreoretinal surgeon, I have a role, and we have to work with a molecular geneticist like Dr. Takeshi Iwata, who is in the basic science, is a photo, apart from inherited retinal degeneration and general genotyping, he's got an advanced lab. And I think that's dream to collaborate with him to have an advanced lab in India too, so that it can work all over India in one place and no need to have replicate the labs, but which is still, we are in the infancy, but the Japan, the National Institute of Sense Organs and Tokyo Medical Center is really advanced, and we have a MOU with them to learn from them. And once we can do the genotype and phenotype, I think with a database, I think we should be able to proceed. The viral vectors for gene transfer, you have the natural virus, you have insert therapeutic gene, remove genes for viral replication, virus carrying therapeutic gene, infect and deliver therapeutic gene to target cells. So our role as a surgeon will come later. I'm glad myself and Professor Kumar Manikvel are working together from 1984, and I'm glad my late father, Dr. N.S. Sundaram, who actually encouraged Professor Kumar Manikvel that time, and also Professor Ratna Savabadi, and started the first genetic eye clinic in Government Ophthalmic Hospital, which is the oldest eye hospital in Asia, and second in the world after Moorfields, which was started in 1819. And virus as a vector. Active clinical gene replacement trials are targeting Stargardt disease and Usher syndrome, retinitis pigmentosa, X-linked retinoschisis, and our friend Dr. Paul Seeming is working on it, who's now moved to the US Davis chorioidermia, where there's a big foundation in US, achromatopsia, and Leber congenital amaurosis, where a lot of my friends in Moorfields are working, and one of them is Dr. Sharola Dharmaraj, who's an ophthalmologist and a geneticist. And these trials use either adeno-associated virus or lentivirus as viral vectors. ABV as a vector for gene therapy, The great advantage in using ABV for the gene therapy is that it possesses minimal immune responses and mediates long-term transgene expression in a variety of retinal cell types. Initial studies have used AAV serotype 2, and nowadays new variants of ABV are being developed to increase the efficiency of gene transfer process. After intravitreal injection, only AAV serotypes 2 and 8 were capable of transducing retinal ganglion cells. Examination of retinal dystrophy patient, you need a detailed history, a detailed family history, comprehensive ophthalmic evaluation, including pedigree charting. There are softwares available, but in our hospital, all the optometrists, the ophthalmologists, the fellows, they are all well trained to do this. And comprehensive ophthalmologic evaluation includes visual acuity, refraction, and retinoscopy, effect of lenses, prisms, or occlusion on visual functioning, tonometry, central versus peripheral visual field defects in confrontation visual field testing, ocular motility and binocular vision assessment, Amsler test, ocular health assessment with slit lamp biomicroscopy. Central and peripheral fundus examination with dilatation, optical coherence tomography, fundus photography, autofluorescence, contrast sensitivity testing, glare testing, visually evoked potentials, electroretinogram, electrooculogram, color vision testing. And Luxturna is a real great hope, which is available clinically. And a Seattle man is the first adult to get a new gene therapy treatment called Luxturna to combat an inherited form of visual loss. In the procedure, which is expected to slow his vision loss but not reverse existing damage, viruses carrying healthy genes are injected into the retina. And the main problem with the public, and I don't blame them, the way they lot of newspapers, news items coming, they all think that any blind can see. And Luxturna, we will just coming shortly to that. Here you have the syringe with viruses carrying healthy gene, and we have to inject into the subretinal space. The main problem is it looks like a simple injection. No, you need a vitreoretinal surgeon. You have to enter the pars plana, go into the subretinal space, and then inject. And here you see the enlarged section of the retina. And this was a dream when we were studying as a postgraduate. We were only studying the 10 layers of retina, and now we have technology to study and as well as document, including OCT and various advanced OCTs. So you have the photoreceptor layer, the bipolar layer, the ganglion cell layer, and you have the retinal pigment epithelium layer where the virus enters the cells, healthy gene released into the cells, and cells return to normal function. And this is how which is working. And you see the gene therapy, the LCA and retinitis pigmentosa with a RPE65 gene. The Spark Therapeutics is the one which has made this Luxturna, and Novartis is marketing all over the world. And I attended a meeting 2 years back in Shanghai when they had a gene therapy on this Luxturna. Choroiderme, X-linked retinitis pigmentosa, Stargardt's Usher syndrome, achromatopsia, X-linked retinoschisis, retinitis pigmentosa, and pigmentosa by optogenetics, and also LCA. And you see the location where they are being done, the sponsors of the program, and where it's registered in the clinicaltrials.gov. An application of gene therapy in different eye conditions, the retinal gene therapy holds great promise, but I think including particularly the patients and the family, should know we are still in infancy, and we will make it. I hope. That's why we have to make the basics right in treating different forms of inherited and inherited blindness. Let's have a look at the eye disease for which retinal gene therapy hold promise. The common retinal dystrophies, the diffuse photoreceptor dystrophies, RP, LCA, and cone dystrophies, and macular dystrophies, Stargardt's, vitelliform, familial drusen, pattern dystrophy, and Sorsby macular dystrophy. Mitochondrial Leber hereditary optic neuropathy is a retinal disease of ganglion cells that leads to blindness in young adults. 1G to A transition in mitochondrial DNA at position 11778 in the gene encoding subunit 4 of NADH dehydrogenase ND4 leads to a position 340 arginine to histidine, the substitution that is responsible for approximately half of the LHON cases. Studies showed that allotopic mitochondrial gene delivery can be an effective and safe mode of therapy for LHON. And here you see the maternal mitochondrial DNA mutation, which presents typically in males in third decade and occasionally in females. And the signs, you see the disc hyperemia, dilated capillaries, like a telangiectatic microangiopathy, vascular tortuosity, swelling of peripapillary nerve fiber layer, subsequent bilateral optic atrophy. Hope one day AI can be incorporated apart from the ophthalmologist visualizing and taking photographs. And Leber's congenital amaurosis in 2008, three groups reported positive results of clinical trials using adeno-associated virus for Leber's congenital amaurosis. In these studies, an AAV vector encoding the RP65 was delivered via a subretinal injection. Initial results from all three studies indicate that AAV is safe in the retina with no dose-limiting toxicities observed. Across three trials, no serious adverse events were observed. Furthermore, patients in all the three studies showed improvement in their visual function and continuing to do well after more than 1.5 years. And you see this is a patient of the RP and LCA. And AMD, currently, wet AMD is treated with frequent injections of recombinant protein into the eyeball. Therefore, scientists are working on long-term disease management by a single dose of drug. I'm glad to say that Takeshi Iwata is also working. We had both, we have trained our Siddhita as well as Dr. Rajkumar, who's a PhD. They both went to the lab. He's working on HTRA1 gene, and also ARMS2 gene, and he also I think we have done a few publications, but we're still in a limited number. Choriodermy, one clinical trial reported in January 2014 used subretinal AAV to restore the RP gene in affected patient. Initial results stated that all patients had better vision, and this is a fundus photograph of chorioderma. And color blindness. Recent research has found out that AAV can successfully restore color vision to treat color blindness in adult monkeys. And juvenile retinoschisis, it is an X-linked recessive degenerative disease of the central macula and affects the nerve tissue in the eye. And the disease is caused by mutation in the RSI gene encoding the protein retinoschizin, important for maintaining the synaptic integrity of the retina. The AAV5 vector containing the wild-type human RSI cDNA driven by a mouse opsin promoter showed long-term retinal functional and structural recovery and improvement in the retinal structural reliability. And this is a foveal cyst in the X-linked juvenile retinoschisis. And everybody knows RP in India because it's widely seen. We have about 500,000 patients of RP and maybe another 500,000 patients of Stargardt's. It's an inherited disease which leads to progressive night blindness and loss of peripheral vision as a result of photoreceptor cell death. And most people who suffer from RP are born with dysfunctional rod cells, so they are effectively blind at night time since the cells responsible for vision in low levels of light. After that, cone cells death occurs, which are responsible for color vision and acuity. Loss of cones lead to full blindness in children. And this is a fundus picture showing the bone spicule-like pigmentation all over retina. And the disease can be inherited as, unfortunately, all the types, the autosomal recessive, autosomal dominant, or X-linked. And when mutations occur in the rhodopsin, the directional protein movement is affected because the mutations can affect protein folding, stability, and intracellular trafficking. Gene therapy to RP is introducing AAV-delivered ribosomes designed to target and destroy a mutant mRNA. And retinitis pigmentosa, RDH11 syndrome, the defective genes are RDH11. This is a normal photoreceptor on the left and mutated photoreceptor on the right. And the another mutation in the rhodopsin structural protein, specifically peripherin 2, it can lead to recessive RP and macular degeneration in humans. There are animal studies which have shown, and if the mutation in the RP is unknown, the gene replacement therapy cannot be administered accurately. And it's again, retinal degeneration is an inherited eye disease characterized by irreversible photoreceptor loss, the activation of the resident microglia secondary to the disease. Stem cell-based therapy has recently made rapid progress in treating the retinal degeneration. It has been demonstrated that transplanted neural stem cells in the retina exerted immunomodulatory effects on microglia, thus delaying the degeneration of photoreceptors. Again, these both, the stem cell and both have lots to work. And this is the work done by Siddhita, Rajkumar, Asta, and myself, and with Professor Kumar Manikwel, screening of genetic variation and different inherited retinal disorder using targeted exome sequencing panel, which was the oral presentation in the 25th IARG-RO in the Arvind Medical Research Foundation, Madurai, where the purpose was to determine the genetic variations in inherited retinal degeneration, highlighting the application of genetic testing and counseling in ophthalmology. We have the RP, the Stargardt's, and Leber kinase laboratories. Results of ongoing project on IRDIs, process flow of genetic testing and counseling, demographic correction, ophthalmic evaluation and possible clinical diagnosis, pre-test counseling, targeted exome sequencing, post-test genetic counseling, and finally re-diagnose. That means you combine phenotype, genotype, and then conclude what is the diagnosis. And these are the results of ongoing project on inherited retinal degeneration. And then here you have a fundus photograph of RP, and then you also have the fundus photo showing bilateral macular dystrophy with fleck-like deposits. And this is a pedigree chart. Proband is diagnosed with rod cone dystrophy. Parents are carriers. Genetic testing reveals mutation in SPATA7 and P.P321 FSTR6 gene. And macular inheritance patterns, the ARMD usually does not have a clear-cut pattern of inheritance, and this is the various clinical trial which is going on. In summary, vision loss from inherited retinal diseases is a debilitating condition. However, with many gene therapies available in ophthalmologists, there is hope for patients. That's why I always quote Charles Keepens, who was the father of modern retinal detachment surgery. He used to say never again to the patient. But I ought to add, don't raise the false hope to the patient, but tell them the realistic. Better they have the genetic counseling, plus they should have a psychological counseling there to understand their condition. And I have a club of patients who have poor vision or no vision, who are my patients either due to inherited retinal degeneration or following retinal surgery or injury. I have a club, and we call Vision Beyond Life. And I have a continuous talk with them, and in the sense we meet. And then they also have counseling for marriage, because most of them are blind. They are upset that they cannot marry. But I tell them, if somebody doesn't want to marry you, so I sometimes wonder whether I'm a genetic counselor or a marriage counselor. But I think it is important for us, an ophthalmologist, who have to take care of the patient and the family. And the main target for ocular gene therapy involves the retinal pigment epithelium or photoreceptors. And the most common method to deliver viral vectors to these cells include intravitreal injection, subretinal injection, or access from the suprachoroidal space. All need a sophisticated vitreoretinal surgeon. The reason I'm telling this is just the word injection. Everybody thinks like a jab, what they take for either a coronavirus or whether they take an injection for a fever. No, this needs a surgery, and you need an advanced vitreoretinal surgery setup with all the advanced gadgets. And these are the references which I gave, and I thank you very much for the patient listening. Thank you. Thank you, Sheila. And I think the next speaker is Dr. Takeshi. So I'm happy I spoke the clinical part. I think it should have been reversed, but I realized it late. But I think Dr. Takeshi Iwata is a molecular scientist who trained and worked in NIH for long. And then he is the head of the National Institute of Sense Organs and also Tokyo Medical Center. And I'm very happy the way the lab is constructed, because Dr. Takeshi Iwata, after his training and experience from National Institute of Health, has created that wonderful setup, which is just a building which is connected by a walkway between the medical center, and just the same level of floor they have done. And everything is there in that lab, right from the cell culture to the animal experiments, everything is there. So I'm happy that we are collaborating with him. Apart from that, Dr. Takeshi Iwata is a great human being, and plus a great host. So whoever is watching this program, you can write to Dr. Takeshi Iwata. And I all welcome you to be, I'm the Secretary General of Global Eye Genetic Consortium. We all welcome you to become member of Global Eye Genetic Consortium. There's no fee at present. we are a member of International Council of Ophthalmology. So we want to spread awareness and create genetic testing, as well as so that one day we'll have a ground where we can have gene therapy, and both India and Japan, because Japan is the land of rising sun and India is full of sun all the places. I hope to work together. And with this, I request Dr. Takeshi Iwata to give some comments on my talk and start his talk.
Thank you very much. A very nice introduction to my talk. I'd like to share the slides.
Sheila, as a co-chair, you have any comments or anything you want to ask before Takeshi starts? Dr. Sheila?
It's an excellent lecture, sir. Are there any questions from the delegates to Professor Dr. Natarajan, sir? Are there any questions? I think we can have the discussion at the end.
Okay. Okay, Takeshi.
Okay. Thank you very much. Thank you very much for the invitation. I'd like to thank Dr. Natarajan and Dr. John for this invitation to the 52nd APAN meeting. I'll be talking about our 10 years of work on inherited eye diseases, mainly retinal diseases. I'm currently the head of the Japan Eye Genetic Consortium and the president of Global Eye Genetic Consortiums. I have done my postdoc at the National Eye Institute at NIH for 10 years, and also at the Bascom Palmer Eye Institute at Miami universities. And I came back in 2000 to start my lab. I got the full floor with myself and the cell biology section, the molecular biology section, the animal facility, the meeting rooms, and the computer servers are all in the one floor. So I'd like to start my presentation by introducing what Natarajan has mentioned. Okay?
Dr. Sheila, you have to mute yourself. Dr. Sheila, you have to mute.
Yeah, sure.
Okay, so this is the structure of the retina, and then these are the diseases from the inherited retinal diseases, including the retinitis pigmentosa, Stargardt disease, choroideremia, Best disease, retinoschisis. We have about 36 different types of inherited retinal diseases. And as Dr. Natarajan has mentioned, that the first gene was identified in 1988, where I went to the postdoc. Dr. George Inanna was the first one to identify a gene, OAT gene, for gyrate atrophy. That was the first gene to be identified for retinal choroidal inherited diseases. And ever since, from that lab, the gene has been increased to about 275 genes. The characteristics of these inherited retinal diseases is that there is a very strong association with the genetics, mainly currently with the association with the gene mutations, which will mutate a protein that is expressed in the photoreceptors or the retinitis retinal pigment epithelial cells. And sometimes it can be on the bipolar segment of the retina. So these mutated proteins is not actually retina specific, but it can be housekeeping genes. But if it can have the mutation, it causes severe abnormalities to the retina functions. These genetics takes in the different types of inheritance. The main one is, of course, the autosomal dominant, which means that you have two genes in two chromosomes, it's the same ones. But if you have one mutation in one of the chromosome in one of the parents, this mutation can be strong enough to give a second generation patient and then go to the third patient. While this is a dominant type, there's also an autosomal recessive, which means that one of the mutation is in both of the parents. So they have two genes, and one of the mutation is in one of the chromosome, and then also in the other parent, also one mutation in another one chromosome, and both chromosome goes to one child. This person will have the diseases. This is called the recessive. Also, there is an X-link. And the X-link, it only goes through X chromosome. So the mother has this mutation, but because she has a normal X chromosome, one more normal chromosome, she doesn't get any diseases. But if this mutated X chromosome go to the male, because male has only one X chromosome, if it goes to the mutated one, you get the disease. So this is going to be a male specific form. This is called X-linked recessive. Now, there's a very unique one called the de novo dominant mutation, which means that this parent doesn't have mutation. The mutation was developed in that patient only and not in other families of the pedigree. So we have this mixture of inheritance for these diseases. So in 2011, we started this Japan Eye Genetic Consortium to study the retinal inherited retinal diseases in Japanese population. Specifically, we started with 6 universities, and now it has expanded to about 30 universities from Hokkaido to all the way to Kyushu Islands. So what we have done is first we set up a database of the phenotype genotype database, and we will start to collect like the fluorescence angiogram, autofluorescence, fundus photo, visual field, OCT, ERG, and many other information, mostly visual, which is very convenient for making databases. And also a pull down menu, as you can see here, that you can just pull down and then minimize the typing. So ophthalmologists are all very busy, so they don't want to spend too much time on every patient's input. So we wanted to make it a very easy way to input the data. And this is how we are working and accumulating data. So up to now, we have collected about 1,500 pedigrees. And out of the 1,500 pedigrees, we have done a 1,200 pedigrees of the whole exome sequencing. And as you can see, 43% is retinitis pigmentosa, the 13% is macular dystrophy, and then you have occult macular dystrophy, Leber congenital amaurosis, Stargardt disease, and so forth. So we're covering a lot of diseases, but mainly it's retinitis pigmentosa in a dominant form, the recessive form, X-linked form, and the de novo form. So what we're doing in the sequencing, the next generation sequencing, is mainly called the short read sequencing. Short read means that the chromosome or the genomic DNA is sort of cut into very small pieces, about 100 to 600 base pairs. And then you sequence this short piece and assemble them into a full genome by a computer. Well, there's another expensive sequence method called the long read, mainly by this Oxford Nanopore technology or PacBio sequencer, which is a DNA goes through a pore and it sequences kilobase, 10 kilobase of long read. Accuracy of this long read is not good, but you get a very long stretch of DNA, which is very convenient when you're compared to when you're doing this short read, because for short read accuracy is good, but assembling is sometimes very difficult. Sometimes you don't know which piece came from where. So that's the advantage of the long read, but this is very, very expensive to do. So for the past 10 years, we have collected and sequenced many pedigrees and identify many genes. So in the x-axis, you have the gene names. And then on the y-axis, you have how many families were affected by these mutations. So the highest one is a gene called EYS. This is responsible for autosomal recessive forms of retinitis pigmentosa. And as you can see, this is like the majority of the inherited retinal disease in Japanese population is caused by the EYS. So looking at this graph, you will know which genes target for the Japanese populations. And I'm sure for different ethnic groups, you will have different genes to tackle. So for India, you have to do this kind of studies to find out which gene is actually most important for Indian population. And maybe because the country is so big that in different regions, you might have a different genes to tackle. So in 2011, when we started the consortium, we only can be able to detect 17% of the pedigree with the known mutations. So everything was unknown, and then you would just return the results saying that, we couldn't find the mutation of your diseases. But now, after 10 years, we are getting to the 53% ranges. So in just 10 years, we have covered quite a bit. And then the other 47%, we need a new technology, which I will talk about in the end of the talk. So we have improved quite a bit. And then 8%, this green part, is completely new genes that has not been reported in other peoples. So these are the new genes that we are actually actively working on. And we are finding new genes every year. This is a gene that we presented in 2020, and in 2021 we just submitted a new gene for glaucoma just recently. And this is 2020. We announced a new gene called for autosomal recessive optic neuropathy. The gene name is MCAT. This is a protein that is functioning as a lipid pathways in the mitochondria. Mitochondria specifically a protein, but this mitochondria protein is coded in the chromosome, not in the mitochondria DNA. It's a chromosome DNA, but it's specific to the mitochondria. So we have this is the pedigree, the recessive family, and we have the phenotype, and then we did all this protein work, and then we also did the animal work. We did the structure simulations, and then these kind of things in combination will tell us that this is the disease-causing gene. So we have been doing these kind of work every day to prove that this gene is a novel. So this success of this Japan IGenetic Consortium was expanded as a global IGenetic consortium. And this was started with a collaboration with the National Eye Institute, especially Dr. Jian Prakash from the National Eye Institute, and myself and Dr. Natarajan, and 4 countries. We started a Asia IGenetic Consortium in 2014, which became a global IGenetic Consortium in 2018. During this time, we had many conferences in ARVO, in Asia ARVOs, APAO, ICERs, and SARC meetings. We had many, over 30 different conferences and meetings, and we will gather with all the local people in this region to identify who is actually working on this iGenetic, who is collecting samples, who is doing the genetic sequences. So those are the things that we have been doing. In 2019, we became the member of the International Congress of Ophthalmology, and we were hoping to expand to Africa and South America through this World Ophthalmology Congress in 2020 and ICER, International Society of Eye Research, in 2020 in Buenos Aires. But because of this COVID-19, we couldn't go. So we lost a great opportunity, but we're gonna build up, maybe starting from next year, we will start to build up this connections again. So through this global IGenetic Consortium, currently maintained by these 5 people, the aim of this consortium is very simple. First is to sort of share information about genetic informations, and then establish accurate diagnostics, establish cost-effective genetic analysis, develop genotype-phenotype database, and also boost this collaboration between this Asia, Middle East, Africa, and South America, which less genetic study is done. And then we will go to these meetings, local meetings, international meetings, to have a meeting like this one. This is the ARVO 2019, to come together face to face and discuss and start the collaborations. So the Global Eye Genetic Consortium now has a publications, a book from Springer Nature. We have finished the 3rd volume in 2021, just came out. Total of 105 chapters from these members. So if you're interested in contributing, we are planning for 2022 publication for volume 4. So let us know. So website is gegc.org. Please connect and become a member. So when I think about the study of this genetic analysis, you start with the patient diagnostic, collect information for the phenotype, you collect the blood or saliva, you extract DNA, do the sequencing, and then you identify the mutation or the gene responsible, and then you do the functional analysis of these mutant proteins. And then you find out the mechanism, how it works, and then you try to make a therapy out of this. And this can be in different patterns. And then you need to start a clinical trial. So in Japan Eye Genetic Consortium, we are still struggling for the number 5 gene therapy and molecular therapy, but we have established moreover about this 1 to 6. But GEGC, we are just starting about number 4 and still trying to collect samples for GEGC. So what is the new technology coming? So till now, we have been only focusing on genes. Genes are regions of the chromosome or the genomics where only protein is coded. While gene is only 1.6% of the whole genome. So 98.4% is unknown. So we are now getting out of the pond and going to the sea, or we were in the planet, but now we're going to the outer space, outer universe. That's the kind of things that we are starting to approach. First, we have to sequence the whole genome of every patient, and of course the controls as well. So many countries have started this whole genome project, including Genomics England, Genome Denmark, and Australia Genomic. And Japan just started last year for the whole genome sequence. So this whole genome sequence will give us what kind of different sequences actually out there outside of the genes. And this is very important to give us the frequency of this genomics. Also, what is outside of the genes? So one of the things that we already know is that although there is no protein coding segment in the outside of the genes, but there are many RNAs that has been transcribed. It's called the non-coding RNA. It's not coding the protein. It's RNA, but it's doing something. We don't know much about these RNAs. So one of the RNAs is called enhancer RNA, and these enhancer RNAs actually work as an enhancer. Enhancer is a region that controls the expression of genes. These expression of controlling the genes actually can be not just downstream or upstream of this enhancer, but it can be very far away from this enhancer, very far away. Also, not just controlling one gene, but it can control multiple genes. So it's a very complicated things that would happen through these enhancers. So we have currently identified 22,000 human retina specific enhancers and also RPE specific enhancers. So I think we will learn from this and then try to find mutation in these regions. The other thing is that through this long read sequences, we are now starting to see that some of the location of this chromosome is actually inverted, inserted, deleted, or sometimes this mobile element, which jumps around this segment of DNA, jumps around the chromosome. What that makes change, this means is that if you have these kind of different sequences, you will have this different kind of chromatin structures. And why is this important? Because if you have this structural changes, it will be influenced by the environmental factors: cigarette smoking, stress, pollutions, UV express, aging, nutrition. All this affects these chromatin structures. So these are still unknown, just started in 2020, but it will be the next generation genomics. And I think we are in a very good age to actually explore this genetic study with inherited eye diseases. So in summary, I talked about the Japan Eye Genetic Consortiums to study the inherited retinal disease in Japanese populations, and also the Global Eye Genetic Consortium to study inherited eye disease with Asia, Middle East, Africa, and South America. And we are looking forward for 2022 to actually meet face-to-face with these people. And I also briefly talked about the next generation, the next generation of the whole genome research. And I'd like to thank all the core members, and I'd like to thank Natarajan as a GZ core members and the funding resources. So thank you very much for your attention.
Wonderful, Takeshi. And as Dr. Sheila said, after the next talk, probably we can have the discussion and question and answers. In the meantime, if anybody has questions, they can write it in the chat, or other participants can raise the hand. So Dr. Sheila, next you can introduce.
Dr. Madhavan sir is here. He has to deliver the lecture.
Hi, Nati. Madhavan here.
Hi, Dr. Madhavan. So Dr. Madhavan is the head of the genetics. Earlier, he was working in Singapore. Now he is doing a genetic project with, I think, 2 NIH grants he has. Maybe he will tell himself. And I'm trying to tell Dr. Madhavan to include me, and he is hopeful, and we are still having some problem. We hope we will do work together. Yes, Dr. Madhavan. You have to tell. You have to...
Okay. Thank you, Dr. Sheila, and thank you, Nati, for this meeting. I think Takashi, we met in Vancouver for the global meeting. I think you remember that we met in Bombay and also in Vancouver. So I'm going to not talk much about my projects now. Today I will be talking mostly about my genetic counseling, how we do it in Nethralaya. Now, the genetic department, we have an arm in this hospital. We do the genetic counseling for the patients whoever we refer to us by the clinicians. The clinicians, there's a separate clinic for genetic clinic mostly by retinal specialists. They identify the patients, and then whoever needs counseling or whoever needs to be tested for genetic diseases, they refer to us. Then they come to us for counseling. We talk to them and make a schedule for the meeting. And we try to tell them what is the disease and stuff like this. So we have good genetic counselors. I've been trained for the past 10 years, more than 10 years, making counseling for the patients. We are also doing this latest technologies for counseling and gene identifications. So the strategy we want to follow is we call the patients, sit for a meeting, talk to them, and then first make them to feel comfortable, and then ask them their queries, their problems, and then we tell them about the disease. What is the disease they have, how it can be managed, and how do we test this disease to identify it, whether it can go to next generation or it will be over in the same generation. We discuss about that to them. And then, if need be, we tell them that they can go for DNA testing. They have to give a blood blood sample, and then they can do DNA testing. In some cases, we don't recommend DNA testing because it's a simple case. In DNA testing comes in money because some of the cases they have to pay money for DNA testing. But some interesting cases, even if we don't have money, they're very, very novel disease. We pay the money from our own projects, and then we get that samples done and then do the sequencing. And once we get the sequencing done or the data analyzed, we call the patients and then tell them what is the result and how do we, we pass all the information to the patient, the information. They come for next follow-up cases, and then they won't go for next testing. The first testing, they may not like to go for DNA testing, but follow-up cases, they may like to go for testing. So we advise them to go for testing, and then we do this entire process of counseling. So basically, we do three things. One is cytogenetics analysis for the patients. Second thing is genetic counseling. And third one is molecular diagnostics. That's what we offer in our department. For each patient, when they come, we take their entire history. They with name, their age, sex, and then come from address, occupation, and then all their details. We put them into the MRD number. We give them a specific number for the hospital. We get the pedigree invariably. Every pedigree is made for all the patients who will come here. And then we get the consent also. We don't get this sample collection unless they give consent for the sample collection. So we make a perfect record of all the information, and then that information goes into the electronic medical records of the hospital. So in case when there's a need for a DNA collection, we try to talk to them. We call them and explain to them the need for the sample collection. They're willing to do that. We collect the samples, blood samples, and then we do the DNA extraction from the samples. And then we do the genotyping by using the NGS platform or single gene analysis platform using the PCR-based screening. It can be a retinal disease, or it can be like, RB1, or it can be mitochondrial genome disease. We do all the sequencing and then do the analysis, data analysis. When we get the data, known gene is affected, we tell them what gene is involved. Many cases, at least 10% of the cases, we do not know what gene is involved. So we call negative, or it's called unsolved cases. Then those genes in this unsolved cases are quite novel for the disease. So getting the data, then we go for genetic counseling and tell them, post-genetic counseling, and tell them what it is, and then tell them what type of carrier the parents are, or how does the diagnosis is going to help them in planning their future child or future disease management. So we have done the genetic counseling for a number of patients, and then as Takashi showed now, RP is a maximum number of patients, because about 60% of the IRD patients are RP, and the second and third are LCA and the Stargardt disease are the next highest numbers. We look at the non-IRD genes. It is RB, retinoblastoma is the maximum cases we get, and LHON and other complex diseases. So entire data for each patient, each disease, we have the records, and so far we have collected about. So this is the number of patients who we have done the genetic counseling and genetic DNA testing. We charge about 6,000 rupees for each patient if we do the testing in-house. I mean, candidate gene testing. We don't send it outside the hospital, so we do it our own lab. It's only 6,000 rupees. But if you send the samples outside for exome sequencing or clinical exome sequencing, it costs about 20,000 rupees for each patient. So some of them can afford to pay that, but some rare cases we pay from our grant and get these things done. So this is our resource in our department. Till 2018, we have counseled about 9,000 patients, more than that, but currently till now it's more than 11,000 patients at present. Of this 9,000 patients, about 7,000 patients gave blood samples for DNA collections, but not all the DNA were subjected for sequencing. So only a fraction of this DNA was sequenced, either exome or clinical exome or candidate gene sequencing. Remaining samples are stored as a depository. We have categories based upon the disease and then based upon the age, sex, and so on and so forth. The entire data is in the EMR records. So anybody interested to collaborate on this any specific disease of from Asian population, Indian populations, it's a good resource. And we have the samples sorted out as South Indian population, North Indian population, Northeast population. So we have got different regions of India have been sorted in different categories. The interesting project, what's ongoing is we have biobank project. That means it is ongoing. We collect samples from the rare diseases or interesting diseases and store the DNA or the blood as a biobank. The entire data in the biobank or in all the records are put into electronic medical records, which is accessible to our staff or our scientists. I just give you a couple of examples of our testing. The first case in RB, we screened for retinoblastoma. The first child was about 12 days old. The mother was affected RB, so she was our patient. So she got married. and then she had a child. They wanted to know whether the child is going to be normal or is going to have a disease. So they came on 12th day of childbirth. So we took the blood samples and then we did the sequencing, and then we found that the child is a wild type. There's no mutation in the gene which normally gets mutated here. It's heterozygous. The father is normal, but the child luckily is normal. There's no RB in this child. The second case is the parents had the first child was affected by retinoblastoma. So they wanted to the mother conceived that they wanted to test before the delivery, check whether the child will be normal or if the child is going to be affected by RB. So we took the CSG fluid. It was referred to us by a medical diagnostic company in Chennai. It's Mediscan. We took the CSG samples and then we did sequencing. And we found that the first child was affected. The second child is normal. Luckily for them, the second child has no mutation. There's no change in the base pair. So a second child was quite normal. So the parents were quite happy to continue the pregnancy. There's another RB mutation screening. So sibling, the one child was affected, first child affected. The second child they brought and then we screened them and I showed them second child is normal. So we do this type of services for the parents, and then we tell them whether the child, in this case, child is going to be normal or child is going to be affected here. So there's the option to terminate the pregnancy or to continue depending upon their decision. So this service we offer for prenatal diagnosis. I just give a couple of examples of IRD gene testing. So, for example, CRB1 gene, we found that both parents are heterozygous. We sequenced them and found that both parents are heterozygous. And then we tested their child, and then it became a C to T transition. C to T transition is a lethal. So this both the child is a patient, it become a disease for the CRB1 gene. So because the transition from C to T is a lethal transition, so it is a lethal case. I mean, genetically it's a disease case. Similarly, we did for PDRE6B. Again, the parents heterozygous and both parents heterozygous, and we found that the child is again the C to T transition, and then we found the child is patient. So we did this couple of examples. So another interesting project what's ongoing now is on the LCA family. How we use the genomics tools for LCA, example LCA as example. So we had about 96 families of LCA patients, and then we took 2 samples from the proband and then the one of the parents as from the family. We did the panel sequencing using clinical exome sequencing. We found that quite a few patients had known genes involved in the disease, but about 17 families they did not show any mutation in the known genes. So they became novel gene variants. So we took the 17 families times 2 individuals each family. We did exome sequencing, exome sequencing, we found quite number of new SNPs and the exonic regions, and then we are working on those genes now. Even after doing this exome sequencing, we found 6 families did not show any mutations in the known genes or known exome sequencing. So currently, we are taking these 6 families for whole genome sequencing. In addition to these 6 families, we also have patients coming to clinical genetic counseling lab for genetic counseling project. When they do the exome sequencing for those patients, this comes from a project, a separate project for LCA analysis. But when we get patients for clinics, we screen them for LCA patients, we screen them for known genes. We found them that they are not showing any mutation known genes. We take them also for whole exome sequence, whole genome sequencing. So from this analysis, we recently published a paper in human variations that we showed mutation in ALS1 gene, which is identified by exome sequencing. And exome sequencing also identified an interesting mutation, new mutation in SLC-CA1 gene in the exonic. And then this is a stop gain function. It stops protein coding function of the gene. And now we are looking at the structure of the protein. How does a normal protein SLCA is behaving? And then how does the mutated protein is behaving? We completed the protein structure analysis, and this manuscript is under preparation. So the exome sequencing gave us one paper recently in this one, and then second paper is being on the preparation for SLC6. This has come out very well because it is a stop gain function, and then we identify the protein. We made the protein structure for the normal gene, and also made protein structure for the mutated or truncated gene. So this is the ongoing project. We work on complex diseases, and then this is the complex pedigree. As Dr. Natarajan was telling, that we have an EndoES project on diabetic retinopathy. We collected the patient a good pedigree, about 60 members in the whole 3 generations. We identified the probands, grandparents, parents, grandchildren, and great-grandchildren who are all diabetic with retinopathy, diabetic with other diseases, and healthy controls. And most of them are consanguineous marriage. It's all consanguinity is known in this pedigree. We prefer pedigrees with consanguinity, and then where we can look for at least 3 generations. So this is one of the complex pedigrees. In this pedigree, we sequence about 30 to 35 individuals, whole genome sequencing of 35 individuals in the pedigree, which healthy controls, diabetic patients, diabetic with retinopathy. Interestingly, all the diabetic retinopathy patients are males. Not even one female was a DR patient in this pedigree. So we sequenced the entire genome of these patients, and then we are analyzing this data, and then we're trying to look at the segregation of which SNP is segregating into the progeny. Very interestingly, the whole genome sequencing data gives us one good thing. A number of SNPs identified in this sequencing data are unique to Indian populations. We compare this with the other ethnic groups, European, Caucasians, African Americans, except one population like non-Finnish Europeans. It is some of the mutations or some of the SNPs are unique to Indian populations. So we are now working with Dr. Sudha Iyengar, who is our collaborator, and then we are trying to make the manuscript for the whole genome sequencing of the family analysis. This is under progress actually. So we also use the genetic counseling strategies for confirming the diagnosis. So some of the diagnoses are made by clinicians, a bit ambiguous whether which one is the correct disease. So we do the genotyping, and then we identify whether this patient is CBD. Similarly, we genotype the patient, and then we confirm that this patient is CSNB. So we can reconfirm the diagnosis of patients by clinicians wherever this ambiguity in the diagnosis. we take it to genotyping of these patients, and then we confirm the diagnosis. We do candidate gene screening in-house. That means if there's going to be a mutation in a single gene, the PAX6 or RP65, RS1 or ND4, ND6, these type of genes, we know where the mutation is located. Then we know the primers. So we make the PCR amplification and then do the Sanger sequencing, and then we clearly identify which mutation they have, and then we can do that testing. So this is another facility we have in-house. So we also do cytogenetics analysis for cases, but nowadays people are more interested to do genome sequencing than cytogenetics. So that technique is not very much used now, but we have facility to do the cytogenetic testing in-house. So this is the cytogenetic facility we have in-house. So we have a facility for array facilities. Some are testing for the genomics. We have the platform. We have the Sanger sequencing sequencer ABI system. We have the fish facility, and then we have the PCR, other facilities in the lab. So we collaborate with a number of institutions, like from New Zealand, from USA, from Canada, from US, and then from Singapore. In Hong Kong, we had a collaboration when Mar was here, Mar Manik was here. Then still we continue the collaboration in-house. And this is our team. We have more people now joined this whole photo. This is our team in genetics department, who are the students and research scholars, technicians, and faculty members. So with this brief introduction, I am trying to tell you the genetic counseling resources we have, and then services we offer in the department. I'm not focusing anything on our research projects, what we are doing in the department. So we have quite a few projects funded by DBT, Indo-ES, MedGenome, Bayer, and ICMR, and DST. So we have lots of projects going on in the department. Each one is on different eye diseases, but I'm not focusing on those projects in this talk. I'm just focusing only on the clinical genetics product. Thank you.
Thank you, Dr. Madhavan, for a wonderful talk. And I think you mentioned briefly about your work with Dr. Sudha. And I think Dr. Takeshi works on IRD and diabetic and ARMD mainly. And I think you are working on diabetes, and as you say, lots to do work in genetic. So I think now we can open the floor for discussion, questions, or any comments, Dr. Madhavan, from your side, what we can do from Madras and Bombay, and along with we have a collaboration with Tokyo, but what we can do within India. And Dr. Kyong is there. VNHA stands for what, Dr. Kyong?
Hello, thanks a lot for your presentation. And I mentioned when I sent you an email, gene therapy, gene diagnosis is very, very new in Vietnam. And I have to try my best to learn from you something. And I have some questions also for Dr. Naradagan, sorry. Okay. Okay. According to you, who is the best candidate for gene therapy? I think that we have many students to do diagnostics, OCT, imaging diagnostic, but what is the thing most important to apply gene therapy, according to you?
No, I think the most important, as I mentioned, is you have to have the, 1 second, I'll just tell you that. See, the most important is you have to know the genome sequencing is required. For example, all RP patients cannot be eligible for gene therapy. So we need RP65 mutation is the only one which will benefit the Luxturna treatment. So any patient with any degeneration, at present we don't have gene therapy available. And particularly I think I'm not sure. India, we are not able to still take part in any of the gene therapy. I'm not sure about Tokyo, Japan. Are you involved in any gene therapy in Japan, Takeshi?
Yeah, we just finished one, and we're doing 3 more right now.
So, can you answer Dr. Chong's question? Like, what is that is important to be done for in a patient? Because he mentioned about phenotype of OCT and other things, but I'm saying we need a gene mapping. Then only I think you can select the patient for gene therapy, because already there's a set.
And Dr. Takashi, I saw you visited in 2015, right? You attend the APAN conference here in Hanoi. And you have any coordinator or branch your consortium in Hanoi or in Vietnam until now?
I couldn't get the last part of the question.
Okay. Do you have any coordinator or any branch of your consortium in Vietnam in Hanoi?
No, but we actually could not connect to anybody in Hanoi. Yeah, at that time, because we always go to these meetings to find 1 or 2 people, the experts in each country. But unfortunately for this EAO or AJAVO, I could not find anybody. So we came back without anybody to contact.
Okay.
So, if you
could not find out anyone, I will introduce you something. I think it's very, very, not international expert, but he or she quite good here in Vietnam. And your consortium do anything like cell therapy or cytotherapy? I mean, that stem cell or something like that. Your consortium. Okay.
So there's many currently not in clinical trial. Well, it's coming to clinical trials. That there are some people in Japan, mainly in RICAN, who are making retina sheet from iPS cells, and they are trying to transplant. But before you transplant to a human, you have to do it in monkeys or something. So currently we're in that stage right now, and yeah.
Okay. I learned some guy in America, they inject stem cell or something like that, subretinal species, and he inject stem cell, but the outcome I think not very good, not very compromising. I mean that, and stem cell I think it's not very good so far. Okay.
Some people have done injection into like iPS cells or ES cells into directly. But that's very rough therapy, and I don't think it's a therapy. It's just a trial. I think it's experiment. I think you have to coordinate into a sheet, because the retina is a very sophisticated, complex tissue. So you have to make it into a tissue, and you can make a tissue out of the cells. So transplant is a very good way of doing this.
And the doctor from India, they talk about Mediscan system. That is an instrument to do the gene diagnostics, right? Mediscan system, like in Sankara, Netaria, the doctor from there, they talk about Mediscan system. Yeah. Do you think that it is a good system to do the gene diagnostics, gene consequencing?
Dr. Madhavan, he has a question. And I put Dr. email ID of myself and Dr. Iwata on the chat. You can make a note. You can write to us for the consortium. So Dr. President is Dr. Takeshi, and myself is Takeshi. I was with Dr. Takeshi in Hanoi. We had a wonderful time in Hanoi for the meeting.
Okay, thank you.
Takeshi, I have one question for you. Next generation sequencing, is it what is different from next gen sequencing and next gen sequencing? You talked about next generation sequencing, right?
Yeah. Well, I meant next generation genomic studies, I think. The sequencing is a short read, but we're stretching. Usually the sequencing, Illumina sequencing is done by about 100 to 150 base pairs, short reads. But currently we're expanding to about 600 base pairs, which is a semi-long sequence, semi-long read sequences. It's much better than the short 200 base or less sequences. It's almost 0.6 kilobase, which is it's giving us a lot of information about structural changes of the chromatids. But of course, if you can do a $8,000 per sample long read, the real long read genome sequences, you'll get a very long stretch of sequence that will give you in one sequence where it's actually inverted or inserted or everything.
One question for you, Ron. Yeah, perhaps the budget for the patient is a big problem or not? Gene therapy. How about the budget? The financial problem is acceptable or quite hard to follow. According to you, which I mean that the income revenue in Vietnam is quite limited. So budget for gene therapy is a big problem or not?
Yeah, so it's a budget is very important. So it comes straight from the government called the AMED, Agency for Medical Development and Therapy. This was established 5 years ago, and it's focusing only on medicines. So you need a big chunk of money to create the facility and also to bring the best people around. So that's quite a bit of money you need.
Thank you.
Takashi, with we have the resources of DNA for different IRDs in our lab. Quite large number of sample size. We are still collecting by the biobank program. We are still collecting the novel patient samples.
Mm-hmm.
Is a global consortium. Is any way we can make a exome sequencing of all the known IRD cases what we have. So any global consortium has any sort of strategy for that?
Mm-hmm. So GEGC has been a little bit struggling. We made a very good connection of people, and however, we are still struggling to get a budget for GEGC. All of our core members have a budget for sequencing their own country people, but it's always difficult to sequence other people outside of the countries. So we have been trying to some of the foundation in the United States and also in Switzerland and to sequence other people's DNAs. And we have just tried to keep on doing this because each of our budget is only restricted to sequencing Japanese, for Japanese population. For people who are in China who only sequence China, for people in Australia can sequence only Australians, and for United States, of course. So maybe we can combine our forces to submit to maybe NIH or WHO or something. I think we have been struggling for this for about 7 years on this, trying to get a budget for GEGC to sequence anybody's DNA samples.
It's not audible. Actually, I think your answer was not audible here. I think some connection problem to the internet.
Oh, I see. So we have been struggling for the budget of GEGC. Can you hear me?
Yeah, but it's not. It is breaking, completely breaking.
breaking up. Oh, okay. Oh, what should we do? So there's a budget.
Ideally, now I will email you, Dr. Takashi. Now I'll email you the that's best of things. So Nati, I'll email you and Takashi. No, join mail.
You will email what Madhavan?
Yeah.
I didn't get you. What did you say about email?
I have the email. I have your email and also Takashi's email. No problem.
Yes.
Yeah.
And sir, Dr. Madhavan, we are planning a global eye genetic consortium after our president's approval, most probably in December, and it will be a grand mega unit, a mega conference. So we want to involve all the genetics in the world, and we want your support. Dr. Madhavan, can you hear us?
I didn't hear you properly, Natarajan.
Oh, what's the problem? Now can you hear? Now?
Go to other room.
I can hear.
You were able to hear me?
I can hear everybody.
They have a problem.
Yeah, I think that room is a problem.
Yeah, just a moment.
Okay.
This is my better.
Anyway, thank you.
Okay, now we can talk.
Okay, so the GEGC has been having problem with the budget problems. So for the past 7 years, we have been trying to get budgets for sequencing of foreign samples. Everybody in our consortium, like me, I get a budget, but I can only sequence Japanese. For my colleagues like Dr. Baird, Paul Baird, he can only sequence Australian people. So everybody has a budget, but they can only sequence their own local peoples. Well, that's the problem with the GEGC, that we don't have a budget for GEGC. So we have been struggling to get a budget and trying to apply to a grant from Switzerland or some foundation, research foundation, RP foundation from the United States, who may permit us to sequence any DNA samples. And unless we get these kind of budgets, it's going to be very difficult to do any sequences. Although I have sequenced like 3 pedigrees from UAE. I have sequenced 5 pedigrees from China. And I have actually analyzed a couple of samples from India too. But that's far we can go with our budget.
Okay, understand. We have small projects for sequencing a group of exome sequencing or group of. But if you want to do large scale sequencing of all IRD patients, who is specific or RP or any one of the projects, any one of the patients. We have more patients, but we have no funding for sequencing the entire individuals. As you said, it's ideal to sequence the whole genome sequencing of all the patients in a given disease, which will give you more power to understand the disease. And then you can compare with ethnic groups like any other populations. So
Dr. Madhavan, can we both our institutes write a project for India?
Yeah.
Oh yes, sure. Definitely we should do that. No,
why not write? Because there is a lot of BIRAC grant now available. So we have a BIRAC grant for clinical trial network.
Okay.
With 6 institutes, we have got about 10.5 crore. I think for genetics, we should do between Shankarnathala and Aditya Jodh. And as an external partner, Dr. Takeshi is always a partner to us.
Yeah.
And we can do a joint project like that. But the grant will be from India for Indian patients. At least 2 institutes we have inherited retinal degeneration as a clinic we are running in Bombay. And we can do it with Shankarnathala. And why not we write a project to BIRAC?
Yeah, we can keep Takashi as as external external advisor or as a resource person, since he's doing lots of this type of work. So
he's already a collaborator with us.
Yeah.
And he also tried JSPC. I forgot.
JSPS.
JSPS. Somehow we are not successful. But it
Yeah.
doesn't matter. We keep applying for even NIH. We're not able, so we can apply all together, and wherever along with the JSPS we can try. And but within India, I think we can have, and we can have him as our as a collaborator with us, and maybe we can't transfer funds to him, but he has his own funds. So that's what is the problem.
I think no need to transfer funds. We can do the transfer. We can do the transfer data. We can, for comparative analysis, we can do sequencing.
That he's ready. See, even he worked with the company. I forgot the name again. Korean company.
Microgen. We're not working with them anymore, but we used to see Microgen. Yes.
Okay. So they had a company.
Microgen is a sequencing company. I think they're quite cheap. I don't know whether Japanese companies can do cheaper, still cheaper. You have your own machine, you can do cheaper, Takashi.
Yeah, Microgen is a pretty cheap, and the quality is not as good as doing in Japan, but it's pretty good.
Yeah, that's so
fast.
Dr. Madhavan, I think we should write a project, and then the same project, Doctor, as you said, ethnic comparison can be done with he has his own funding, and then in case we have a joint grant from NIH, that also we should apply.
Yeah, I think maybe you can nominate one of Sir Rajkumar or somebody. We can.
No, Rajkumar has left us. We have Siddhita. And I was just telling Dr. Takeshi in the beginning before you joined that now we are having an MOU with the National Biotechnology Lab with the Khalsa College. They have an excellent lab, but they're not into genetics, but they are into molecular biology. So I'm actually trying to do with them so that I will shift the lab here. And we can, Siddhita is the person. She's right now in, and we have 2 MSCs now.
Okay.
So I'll connect with, and I don't know whether they're not logged in today, but I think we have a YouTube, so we'll connect with them.
Okay.
And probably then once we are strong, then we can help Vietnam and other Asian countries.
Probably we can take the help of Takashi to design the project, which number to calculate the power and things like this. Probably he may help us to design the project, and then we'll try to get funding from Indian side, and then share the data with him for global consortium.
No, both Siddhita and Rajkumar has worked under Dr. Takeshi in Japan. So definitely we can prepare the basic. Then we can work together and send it to Dr. Takeshi. We'll do that.
Okay. Yeah, sure.
Hello.
Good. So it's going to be 3 minutes more, no, Sheila? Any comments from you? Actually, we should be able to do a lot of tele-ophthalmology with genetic eye diseases in India, both Shankarnetrala and Arvind and Aditya Jyot. So why not you take up the tele-ophthalmic clinic and put Madhavan there?
Yeah. Genetic counseling is through tele-ophthalmology very effective.
No, not only genetic counseling, even the exchanging of the fundus photo, OCT, and patients are going from pillar to post because nobody guides them in the sense patient is expecting some dramatic treatment, but we have to tone them down to understand what is the disease, and we say that we are all working. There may be some rich patients like America, the rich patients give money for the grant. So we can try to get grant from the patients also. Because RP also, there's a big group in India.
Yeah, a lot of RP patients are there.
No, at least we can do a joint clinic, I'm saying. Tele-ophthalmology clinic for genetic eye diseases.
Yes, that can be done.
Yeah, or we can have a joint program with Shankarnetrala for the public. We can have an introduction for creating awareness.
Yes, sir.
Using we can use the under TOSI and what is the other one? What India? Echo India. So they can address it to the public. And it has to be, the lecture can be toned down to public. What to expect. And Dr. Takeshi gave a wonderful public talk here in an oration about 3 years back. And also Professor Kumar was there that time.
Yeah, we can take it up, sir.
Okay. Thank you, Dr. Madhavan. Thank you, Dr. Takeshi and friends from Vietnam.
Thank you.
Me and Dr. Takeshi have enjoyed the Vietnamese food.
Yes.
And I had it spicy, and he had a non-spicy.
Yeah, a lot spicy. Indian food. Yeah, so much spicy inside. But I like curry worldwide food curry. Yeah. Thank you.
Yes, yeah.
Thank you, sir, for participating.
All of you are welcome.
Hope see you again.
All of you are welcome to India.
Yeah, thank you very much, and see you later.
Yay.
See you later. Yeah.
Bye.
Bye-bye. Thank you.
Thank you, Takeshi. Have a nice day. Thank you, Dr. Sheila. Thank you, Dr. Madhavan.
Bye-bye. Bye-bye, Natarajan.
Bye. Thank you.
Vân Anh nó cảm ơn rồi nó tán thưởng, nó gửi cho mình chứ không gửi cho everyone. Bà này cũng ít đi hội nghị, cứ biết gì.